The material is just way too intertwined to say that it's either for lecture or lab. So So what that means to you is that on exams or questions, you could see material from this lecture on laboratory and lecture exams and assignments. Yeah?
Sorry, Professor, before you go on, are all the videos up from last week's lecture?
They should be. The Tree of Life one is...
It's only an hour and 23 minutes. I feel like it should have been like 2 hours.
Well, each class is 50 minutes, and I know at least on 1 or 2 of the classes I ended early a little bit so as not to begin a new topic, only to end it after a few minutes. So if I haven't, if you look at it and it doesn't have all of the material in it, let me know, but I think I've posted everything. All right. So 16S rRNA. In this lecture, you're going to see a few slides that you've already seen before, just to remind you about the things that we've already talked about and to make sure that everyone's on the same page. Oops. So, one thing that I've explicitly stated and alluded to multiple times is the fact that most of the things in the environment we can't grow in a traditional laboratory setting. Like the organisms in our rhizosphere, we'd probably have a difficult time growing 95% of them. And this was known quite a while ago, and the way that they figured this out was what's called through the Great Plate Count Anomaly. So if people went into the environment and collected some type of environmental sample, and they would culture it, and they'd count the number of cells that they would be able to grow. However, if they took a sample from that same environment, looked at it under the microscope, and counted the number of cells, what they found is that the number of cells they could count was much greater than the number of cells that they could culture. So there were all of these cells that somehow they weren't growing, they weren't being cultured, and for a long time those cells were completely inaccessible for study because they didn't have PCR, DNA-based methods for studying these. So the way, of course, that we can start to study those cells that we can't grow is through DNA-based work, which is what we're doing in here, in our culture-independent portion of the lab. So again, we're going back to the ribosome, this magic piece of machinery that is so well conserved across all taxa that allows us to do a lot of these studies. And this is a similar picture to what you've seen before. I'm just blowing it up a little bit. And as a reminder, the ribosome is made up of these 2 subunits, 50S and 30S. And I've gone over this, so I'm only going to briefly hit on it. But S refers to the Svedberg units, referring to the velocity when centrifuged. So remember we talked about you can put these in a very viscous solution, you can centrifuge it, and the distance that it migrates down in your tube is related to the mass. So things that are bigger will migrate faster. And so they assigned these units to tell us how fast it would go down in the tube. And the key thing to note is that there are a bunch of proteins, but there are also a bunch of RNA, pieces of RNA that never get translated into protein, that these RNA themselves act as just key components of the ribosome. And from here on out, we're going to be talking about the 16S rRNA sequence that is part of the small ribosomal subunit. So this is the one that we've chosen for a number of reasons to use for most taxonomic studies. Questions on this? Just covered it briefly since we've touched it before. Okay. So there are several reasons why we use our RNA sequences, and of course, one is that they're universal. We've talked about this again and again across all species in all domains of life. They're highly conserved functionally, and because they're performing the same function in all different organisms, the sequence and the two-dimensional structure, three-dimensional structure is maintained. Now, one of the things that makes it so great for this type of study is that there are some conserved regions and some variable regions. And hold on to that. I'll show you a picture of what that means exactly in a few minutes. But this is one of the big reasons why we are practically able to study these genes. And then importantly, horizontal gene transfer is rare. So what is horizontal gene transfer?
Conjugation.
Yeah, conjugation, which is?
Bacterial sex.
Bacterial sex, which is?
Sex pilots.
Yeah, so what is the effect of this? Yeah.
Transfer.
You're transferring DNA from one bacterium to another. And so if, and some genes are pretty promiscuous, they can go from one cell to another, to another, to another. And as you can imagine, that would mess up any phylogenetic information that you would have. So if you were trying to figure out what species a gene comes from, and this gene kind of hops around to a bunch of different species, that's not going to give you good information. But since horizontal gene transfer is rare, and the ribosomal rRNA genes aren't moving around to different species, this is a much better indicator of what taxonomic group they're from. And again, a reminder of just how conserved some of these sequences are across all groups of life. Really similar. This is represents the structure of the rRNA molecule. So this is the two-dimensional structure, and then folded up is the three-dimensional structure. And what becomes really important is that the rRNA form these hairpin loops. And this is going to be important this is important to the whole systematics of why we can study rRNA and use it as and why it's such a great taxonomic marker. So let's say that you just have let's draw a picture of a hairpin structure up close. Okay, or a hairpin loop. So here's our hairpin loop, and these are the base pairs. And this forms, these nucleotides are base pairing and around the stem, and then in the loop, you have nucleotides that are not base pairing. So there's no base pairing among these. These are just kind of free floating. So if you were to add in the sequences, let's say this is an A, C, T, T. Let's say this is a G, G, A, T, C, C. Just based on, what would this have to be? What would that have to be right there? Yep, A. and that would be an A, G, and then that would be a T. So you can see that the sequences here are constrained, because if let's say A needs to base pair right here, and let's say there was some type of mutation. So let's say there was a mutation, and I'll just draw a small part of it. So this would be a portion, that would be our C and our G, and over here would be our T. And let's say this mutated to a T, then you no longer get base pairing. So these sequences here are very constrained because the mutation is going to really mess up the structure of the base pairing in these hairpin loops. Whereas if, say, this were to change, it wouldn't matter nearly as much. So if you look across this entire two-dimensional structure of this rRNA molecule, you can see regions that are base pairing, and those are most likely to be conserved. And regions where there are the loops, and those aren't as constrained. Those are a little bit more free to change. So what does that mean in practical terms? Or what do we do with this information? Well, one of the things we do, of course, is ask who is there. And you'll hear this phrase over and over again, who's there? And let's say that we have, this is a close-up of our microbial community. In our case, it would be in the rhizosphere, and you have a bunch of different cells, and they have circular genomes. And in this case, I've colored the genomes by the species that I have made the picture to be. So orange, everything with an orange genome is a particular species, everything with a red genome is a particular species, and so on. So we extract the DNA from the sample, and this is exactly what we did on Wednesday. We took our soil, we took all of the organisms in the soil, and we just extracted the DNA. No worry about growing or anything like that. We just got all of the DNA out. And on each genome, each organism will have a 16S rRNA gene. And you can see that I've drawn that 16S rRNA gene on each of those genomes. And we can PCR amplify that, and we'll go over that in a second. But we can amplify using PCR that gene, and what we end up with is a PCR product, and our PCR product is from all of the different organisms in our sample. So we have, and the number of different times we see a particular PCR product tells us about the abundance of each organism. So in this case, we have 3 orange, and so we get more pieces or more PCR product from the orange ones than we do the others. So from this, we are able to get abundance and information about who's there. And we can then use the sequences that we generate to compare them to different databases, and we're able to make a functional assignment. We're able to say, okay, orange, if we look at a database, it's really, really similar to something we already know, so we're going to say that it's from that particular organism. And again, this is exactly what we're doing for our rhizosphere project. So what about the 16S rRNA gene makes us able to do that? And it goes again to our variable regions and our conserved regions. So let's just say that purple is, or actually, no, we don't need to do that. Okay, lime green are our conserved regions. So I'm going to, in here, kind of circle some of our conserved regions. And again, those are going to be our stems. And you can see up here that we have in our diagram of the 16S rRNA gene, we have these lime green regions that correspond to this two-dimensional structure. So this is what it looks like on the genome. Of course, anytime you have a gene, it's just this linear, it's a linear piece of DNA, and the transcription machinery goes along and it makes mRNA. And the mRNA is spread out like this. You have conserved variable, conserved variable, conserved variable. Yeah?
Sorry, I'm a little confused. So conserved is supposed to be like the places that are not going to be changing?
Right.
So why did you circle those places that are not pairing up?
I circled the stems, the places where there's base pairing.
Oh, okay.
Yeah, I guess a circle was probably a bad thing to use there. Let's see. I have an eraser somewhere. Is that my eraser? There we go. Well, the biggest variable regions are the 9. So there are little small ones throughout the rRNA gene, but the big substantial ones, there are 9. Okay. It's not letting me erase, but just to note, so when I put this circle here, I guess I should have put a box that indicates that anything I have in green are the ones that are base pairing. They're forming the stem. Whereas in gray are the variable ones, and you can see here you got some major variable regions. Those are the ones that are freer to mutate, and those are the ones in gray. So does everyone understand how this linear representation of the DNA corresponds to this two-dimensional version of the rRNA structure? Does that make sense? Variable, conserved, why each piece is variable and conserved. All right. So the reason that having conserved regions is so important has to do with amplification. Now, how do you think that we or what is the first step whenever you're doing a PCR amplifying something? Is everyone pretty familiar with PCR? And you know what I mean when I say primer? Yes. Okay, everyone's nodding, okay. Anyone not? Anyone need a little bit of a primer on primers? We're good? Thank you. Some of the only laughs I get are my terrible puns. Okay, so they are. Okay, so when you're designing primers, in order to amplify some DNA, the primers need to match exactly the location that they're targeting. So what would happen if I put a primer in this V4 region? Would it amplify everything from my soil sample? No, it wouldn't amplify everything from the soil sample because this is variable. So this primer would match the region from some species, but not from other species. So we wouldn't get amplification from all of the species in our sample. However, if I made a primer that matched that region right there and that region right there, those are conserved. Those are really conserved. So these primers are considered universal primers and will amplify, you can never say all, but will amplify most species in our sample because it's so well conserved that things aren't going to be, there aren't going to be differences here at all. And so the primers will match everyone. So you can use those primers, you can do a PCR, you can amplify, and you get DNA from everyone. And that's what makes the 16S rRNA genes so powerful, is that there are these regions of conservation, and in the middle, there are these areas of variation. Because we talked about what it would be like to try and build a tree when all of the sequences are the same. It wouldn't work because you don't have any information. You can't build a tree if everyone is AAAA. So having this variable region that's flanked by these conserved regions makes it possible to do these analyses. You get a piece of DNA from everyone, and then you can use this variable region to build your trees. Now, when I say variable, does it mean it's super variable? No, it's not. It's actually not super variable, but it varies enough that we can build our trees from it. If it were really variable, like if it changed really quickly, then it would be almost like having a random sequence, and that doesn't do us much good either. If you just have some, someone just putting up random pieces of DNA. But because there are just some changes, it makes it so we can build our trees. All right, so again, you see this same diagram of our soil, and we have extracted the DNA. And the next step in the lab process will be to do PCR. And here I have a link to a video on exactly how PCR works. For now, this is for your reference. When we get to this step in the next week or so in the lab, we'll actually go over exactly how PCR works. We'll watch later in class. So for now, for the purposes of studying, just know that we do the PCR, and that eventually we'll get to the nitty-gritty details of how PCR works, but not for the purpose of this lecture. All right, and again, we design our primers. In this particular case, in this example, the primers are sitting in these conserved regions, and the piece of DNA that we get is all of these pieces right here, and we can use the information from all of the variable regions to build our tree. And once we have our PCR product, again, once you do a PCR, all you have is a little tube of DNA still. You need to go on to another step. After you do your PCR, we can proceed to downstream applications such as DNA sequencing. And what does that look like? So let's say now we have done the DNA extractions, we have done PCR amplification, and we've done sequencing. So now we have, they give us what you end up with after you sequence at the very end is a text file of your sequences. So I represented that here by different colors. So this is species orange. And in reality, you'd have a text file saying, well, this is, A, C, C, T, T, whereas in your file that you get from the computer, the blue one might be A, C, G, T, T, and so on and so forth. I mean, just like the sequences at the beginning of the assignments that you've been doing. Okay, so now we have those sequences. We still don't know who's there, right? I mean, our end goal is to know who's there, and this isn't telling us who's there. So what do you do next? you compare to a database. And you're going to hear me refer to databases over and over and over again. And one of the really neat things about science is that there's this enormous focus on having everything be public and open source. And that's one good thing about having the government fund science, is that everything is publicly available. So before, actually I'm in the process right now of getting a paper published. And before it goes to publication, all of my sequence data, everything that I've generated, has to be uploaded to a public database so that anyone else in the world can access those data. And that's really important because the government has funded that. And collectively, it increases knowledge. So if you have all of the scientists who are doing this type of work, putting their sequences into the database, that means that someone else can take the sequences that they've generated and compare their sequences to my sequences and everyone else's. Does that make sense? So we've got these big, they're great, these databases. And we can, this is what we're going to do. When we get our sequences back, we're going to compare our sequences to a big database that other people have generated. And what we'll be able to do is make taxonomic assignments to our sequences. Now, the databases are enormous and awesome, but they aren't perfect. So you might have some where you're able to say, wow, this is exactly the same as this particular species. And we can confidently say that that piece of DNA is from that particular species. However, the environment, especially in soil, the microbial world in soil is still super diverse. Still, there are so many unknown organisms in there. There's a decent chance that you're going to have a PCR product, a piece of DNA, that doesn't match anything in the database. I mean, we're still at that point in this part of microbial ecology where we still don't know what's out there. How many of you have heard these recent stories about them resurrecting a bacterium from a mine or cave recently in New Mexico? Anyone been hearing the news stories, too busy studying? So,
and it happens so frequent. I've heard about it from other places, from glaciers and stuff.
Yeah. Well, so the latest news story is that people have isolated that in these big salt caves, I think in New Mexico, could be somewhere else entirely, that there are these spore-forming, dormant bacterial cells. And the latest claim is that they've gone into these environments and they've been able to have the cells germinate and grow them. So everyone's saying, wow, this is so cool. These are 50,000 years old and they don't look like anything else we've seen. That's pretty cool. And it sounds really cool to the general public. But the thing is, I can go out and sequence a whole bunch of dirt and sequence a ton of stuff no one's ever seen before. So yes, it's cool. However, there's so much out there that we don't know about that it's not as novel to sequence something new as the media would have you believe. So my point is that when you do this type of thing, you're going to get some that match things in the database, and you're going to get some that you don't know a whole lot about. Some, like you may find that it matches pretty well to the Geobacter genus, but there aren't any species in the database that match it exactly. So your particular sequence, you know the genus fairly well, but no one has isolated that species before. And from that information, you can build your phylogenetic tree and look at the relationship among the different sequences that you have. And again, we're going to be doing this in the lab. Now here comes something that's a little bit tricky. And there are a lot of, even microbiologists, depending on what type of microbiologist you are, it's not something that everyone has kind of their head wrapped around, I guess I should say. And the a big question right now is how do you define a bacterial species or an archaeal species? And defining a species is pretty simple when you're looking at you've got humans, you've got chimps, you've got gorillas, or you've got elephants, or you've got all of these different things where it's pretty easy to say the delineation between species is fairly easy. And traditionally, delineating between bacterial species hasn't been too tough either, especially in the age where people were most interested in disease-causing organisms. So Mycobacterium tuberculosis is an example of a species. Clostridium tetani, or whatever that is. Tetany. I should have taken Latin. So that causes tetanus. Any other types of species that you can think of are considered species. So no big deal, right? Bacterial species. And people classified these species by morphology, functions, and to a large extent that's often been disease, and DNA. However, with the advent of this type of 16S sequencing has come a problem, like how do we define a species? Because what happens is that you don't have any information for a lot of these organisms, you don't have any information about what their functions are. You don't have any information about morphology. All you have is some DNA sequence from the 16S rRNA gene. And to define a species, you have to have some type of cutoff. You have to say, well, how similar do two sequences have to be in order to call them a species? So let's say you have your environmental sequence. So let's say this is from this particular sequence, let's say is from Cabernet Sauvignon sequence. So from one organism. And then let's say you have your database over here with all sorts of different types of, all sorts of different species. Now, how identical does this have to be to any one of these? Of course, if it's 100% similar, then you can say, yeah, it's probably a species. But we know that there's variation in species. There's variation in the DNA in between all of us, so species can vary. So what happens if it's 99% similar? Is that a species? Is that the same species? What happens if it's 95% similar? Is that a species? For reference, humans and chimps are about 99% similar. So what about 90%? How do you, yeah?
Isn't it about the expression of the genes? We can have the same genome, but it's about the proteins that are being expressed. Is that how you're looking at it, if there's someone?
No, we're just looking at sequence on the DNA level. Because remember, all we have is information about the 16S rRNA gene. And we want to know, are two things the same species? Or let's say that, for example, you've got some type of diseased plant and you want to know maybe if there's a disease-causing bacterium that's living in the roots. So you sequence the 16S rRNA gene and you try and match it, and you get something that's 98% similar to a known disease causer. Is that the same species? Yeah?
Is that like for the whole genome or just like key points?
That's for the 16S rRNA gene. Yeah, because right now that's all we have the information for right now is our 16S rRNA gene. So, what is the same species? Well, what people have decided by doing a large number of studies comparing different genomes is they have figured out that about 97% identity, or well, let's just say similarity, is the same, and we're going to put this in quote here, species. Because, yeah.
So if it's 16S, is like how you consider it? Can you just compare the variable parts then?
Yeah, that is the variable parts. So yeah, so in the variable region of 16S, if, say, this particular sequence is 97% similar to this, then you would call that a species.
Same thing.
Well, here I was just putting down numbers like, what if? This is hypothetical. What if it was 99? What if it was 95? What if it was 90? So people have come up with this common way of saying things are the same species, and that is if they're identical at 97% identical. So if you have, what that means is if you have 100 nucleotides, that means that 97 of them have to be the same, and 3 can be different for them to be the same species, which is a pretty low threshold considering that humans and chimps are the same at 99% of nucleotides. So in terms of bacteria, we're being much broader in our definition of species. And to denote the fact that when we're comparing a 16S sequence to the database, we don't have other information like what the cell morphology is, what types of function it performs. To account for that, and to account for the fact that we really don't know if they're the same species, we've adopted a different term. And if you ever encounter a microbial ecologist, they're going to be very hesitant to say anything is a species. You'll either see them put it in quotes, species, or you'll hear the term OTU. How many of you heard that, OTU? No? Okay. So if you talk to me, if you talk to Dr. Flores across the hall, and depending on the context, Dr. Cooper just over here, you'll often hear us refer to OTUs instead of species. Though if you talk to more traditional microbiologists, such as Summers or Barisi, they'll refer to species much more easily. But those of us who are environmental folks and look at communities will refer to as OTU, operational taxonomic unit. So that's our way of saying, yeah, it's pretty close to what we think is a species, but instead of a classical definition of species, we've defined that as being different or similar at the 97% similarity threshold. So it's just the fact that we've defined a species as a certain way or an OTU as a certain way, and so that's what we're calling it. If you ever hear us refer to a species, then it's going to be in a more traditional context, like Mycobacterium tuberculosis. So, does that make sense? I see some puzzled looks. It's not necessarily an easy thing to wrap your head around, and I go about my daily life thinking of OTUs as an amorphous species. It would be kind of like if you crossed a human and Neanderthal, which did happen, by the way. And it's kind of a species, a little bit similar. Okay, well, we'll come back to this because this is so important for the class. We'll touch on it again and again, and hopefully by the end of class you'll kind of have your head wrapped around it, because you will have done, actually, you'll be working with these data. Okay, So here's some text written out of a lot of the things I just talked about, mostly for your studying convenience. But again, a common way to define a species is if they are greater than 97% identical. So what's kind of interesting is, this goes back to the fact that 16S is really well conserved. So you might have two 16S sequences that are 97% similar, but the genome can actually be more different than that. And what that means is that, let's say you have a genome here and a genome here, and we'll have our 16S be right there, and you compare these, and let's just say that they are 97% identical at the nucleotide level. So we call them an OTU. Essentially, we've grouped them together in a species-like group. But these are super well conserved. So it may be that the rest of the genome, so let's say, just going to put dots around that, these only maybe, say, 93% similar, because you don't have the pressure on the rest of the genome, the extreme selective pressure, as you do on the 16S genes. So we're getting to the point where our definition of a species is even looser than we had originally thought. Okay, and again, the concept of a bacterial species is somewhat amorphous. Okay, so I'm going to end the lecture there. Oh, that's a little smiley face. I almost can't draw a smiley face if I tried, but it's an accidental.
It's like an owl.
Yeah, it could be an owl.
Like 93%, like eyes like looking up.
Yeah.
16S are here.
Okay, but anyway, we'll end there.